So let's get started. I think I'm recording. Double check. Yeah. So today we're going to finish the carbohydrates chapter, chapter 8. And we're going to do the last section, which is about glycoproteins. At the end of class, I already announced, but in case no one heard, I have your exams for you. So if you want to look at your exam ones, see what you got wrong, feel free to take your exam. The answer key is posted on Brightspace. And what else? Oh, and tonight your quiz 6 is due if you haven't submitted it already. And that's pretty much it. Yeah, so we'll finish chapter 8 this week. Next week we'll pick up chapter 9 and 10. And then your exam 2 is 2 Thursdays from today, I believe. Time flies when you're learning biochem. So let's start by doing a little bit of an overview of glycoprotein functions. So there are 5 descriptions written here on the slide and only 3 glycoproteins. So please use all of the descriptions and match them to each glycoprotein. So each of 1, 2, 3, some of them will have more than one answer. And take a few minutes to match those up. Does it sound kind of broad or vague? Yeah, so proteins can be secreted, or they can be integral to the membrane or somehow attached to the membrane. Is that where you're hung up? Like secreted or membrane? Okay, yeah. Oh, like a protein is secreted or membrane. Yeah, that's kind of, yeah, more words necessary. And technically, you could say membrane proteins are secreted proteins, but just trying to be explicit. Okay, so does anybody want to start us off? Which one of these or more of these descriptions matches with peptidoglycan? Yeah. C, yeah. Peptidoglycans are very important for helping bacterial cell walls stay rigid. Excellent. What about proteoglycans? Not to be confused with peptidoglycans. Yeah. Yes, it is a component of cartilage. Is there anything else about proteoglycans? Yeah. Yes, it is also a highly hydrated and flexible structure, which is what makes it so great that it's part of cartilage, because it helps to cushion your joints and stop your bones from creating friction by rubbing against each other, which would not be fun for anyone. Okay, so that brings us to glycosylated proteins. Which of these describes glycosylated proteins? Also process of elimination. Yeah. A and E. Excellent. So glycosylated proteins, A, blood cell antigens that determine blood type, are a type of glycosylated protein. They're carbohydrate antigens. And also E. So glycosylated, most secreted or membrane proteins are glycosylated. They form almost like a sugar, or a lot of membrane proteins that are glycosylated form kind of like a sugar coating around a cell. You can think of it like a candy coat, like an M&M. Every cell is an M&M. How's that for a mental image? So yeah, that's what glycoproteins are doing. So let's now move into some of the individual glycoproteins. So first we're going to look at peptidoglycan, and we have a diagram of what that looks like up on the screen. So everybody take a few minutes and label the components of this structure, and then we'll go over it together. All right, so before we get into labeling the specific parts, can somebody tell me which parts of the peptidoglycan are sugar and which parts are protein? Yeah. Yeah, exactly. So the yellow and the orange are sugars, which leaves the other parts, the bluish green and purple parts. These are protein. Does anybody remember the names of those sugars? Kind of a mouthful. Yeah. Yes, exactly. And I won't write them in because I have the labels popping up in a sec, but those are the sugars. And then what are the two protein components and which is which? Or even if you're not sure which is which, what are the protein components? Yeah. Yep. Exactly. Yeah. So all together, we have our sugary part. That's that yellow and orange alternating molecules that are kind of linear. N-acetylmuramic acid and N-acetylglucosamine. And then those kind of chains are connected together with peptides and little bits of protein. So we have a peptide chain. That's the little bluish, greenish circles. There are 4 of them. And then there's the pentaglycine bridge, which, as the name suggests, has 5 glycines in it. And those are purple, and they connect together, again, those linear sugar molecules. And the pentaglycine bridge is this purple one that's kind of horizontal. Yeah. All right. Any questions about peptidoglycan? Okay. So, and then I guess before we move on, this is peptidoglycan. We really don't want to confuse it with proteoglycan, and we're going to go over proteoglycans now. So, names sound very similar. They are actually very, very different things that are found in very different places and have very different functions. So, make sure you know which is which. So that being said, here's a proteoglycan molecule. So take a couple of minutes, label this one, and then we will also talk about it. Okay, so that should have been enough time to work that out. So yeah, so let's, I guess, start again by figuring out which part of this structure is made of protein and which part is made of sugar. So which parts are made of protein? Yeah. No, so not to give it away, but the blue and green are actually saccharides. Yeah. Yeah, this part is protein, and pretty much the rest of it is carbohydrate. So, and I think there are also other components that are not drawn on here. This is slightly simplified, and it does have other, I think, protein components, but you don't have to worry about those. So, so that being said, what are the other parts of this figure? Anybody can name any piece. How about this long kind of red line that goes down the middle? Yeah, it's not fun to say, but yeah, that is hyaluronate, and I have the labels popping up in type so you don't have to read my handwriting. And then, yeah, what else do we have? What are like these components here that are shown in green and blue that look kind of like brushes? Yeah. Yes. Chondroitin sulfate. Yeah. All of these terms are a little bit of a mouthful, but it's fun to say. Um, so, how about these little smaller black lines that are right underneath those keratin and chondroitin sulfate molecules? Yeah. Yeah, those are end-linked oligosaccharides. And then one other component that we didn't quite name, the protein component, is called, the core protein. Yeah, easiest one to say. So that is all those names in case you're not sure how to spell them.
Keratin sulfate and chondroitin sulfate, those are O-linked, right?
Yes, they are O-linked, and they are also, they're both glycosaminoglycans. Cool. So how does this very odd-looking structure relate to this molecule's function? So we talked about what it does, right? It's a part of cartilage. So how does it help us out by being in our cartilage? Yeah. Mm-hmm. Yeah, exactly. So those types, this whole thing in general, it's full of all these molecules that are charged and polar. All of those love interacting with water. So these types of molecules are really, really good at taking up water. Like I think hundreds or thousands of times their mass in water, like a lot. And then they have this characteristic that they're compressible, so you can squeeze them, and some of that water will come out. But they're also elastic, so because, like I said, a lot of the molecules or components in here are negatively charged. If you compress them too far, those negative charges will start to repel each other, and so that's what gives it this other characteristic of elasticity. So you can squeeze them, water comes out. Squeeze them too much, they bounce back. And they'll take in more water as they re-expand. And so this is a really good way for them to be able to expel out waste when you squeeze them, and also to bring in new water and new nutrients when they expand back out again. Because they're also found they are found as part of the extracellular matrix, and so they're not inside of a cell. They have to get nutrients some other way, right? All right, so that's radioligands. So now let's talk more about glycosylated proteins in general. Or just types of glycosylation. So everybody can take a few minutes and think about O-glycosylation versus N-glycosylation, when it happens, where it happens, what's happening, etc. And we'll discuss it together in a few minutes. Okay, so let's start filling in this table. So let's start with when we add these types of glycosylations. So at one point during protein synthesis and trafficking, does an O-glycosylation get added? Yeah. Yeah. So for O-glycosylation, it is post-translational. And so that means it's happening in what organelle? If it's already been translated. Yeah. The Golgi apparatus. Yeah. And so that is in, yeah, let's just keep going with O-glycosylation. So what what are we adding when we start off an O glycosylation? Like how many sugars or what type of sugars? Go for it. Yeah, so often it's galactose plus N-acetyl galactosamine, or it can also be N-acetyl glucosamine. Depends on the glycosylation. And so how many sugars is that? 2. Yeah, so it's always 2. Or a disaccharide. And so when we're adding on this disaccharide to a protein, what amino acid are we adding it to? Yeah. Yeah, excellent. Serine or threonine. And this makes sense, right? Because serine and threonine both have hydroxyl groups, and we have an O glycosylation. So we need something with an oxygen to add it on to. All right, so that's O glycosylation. Now let's think about N glycosylation. So when does N glycosylation start? Like at what point during synthesis and trafficking? Yeah. Yeah, it starts co-translational. And then it continues post-translationally. So since it's co-translational and post-translational, what organelles are we seeing this editing happening in, adding and editing? Yeah. Yeah, it starts in the rough ER. And does it continue on anywhere else? Yeah. Also the Golgi. Okay, so what are we adding on when we're adding an N glycosylation, when we're starting it out? Yeah. Yeah, it is a 14 sugar oligosaccharide. And as it goes through the editing process, we're going to end up subtracting and adding a number of sugars. So you start with 14, but you will not end with 14, usually. Or sometimes you do, but not always. So what amino acids, then, are we adding this 14 sugar oligosaccharide onto? Yeah, asparagine. And do you remember the sequence that you need to add it on, aside from asparagine? It's okay if you don't. Anybody? Yeah, exactly. So you need asparagine and then X, where X is any amino acid except proline. And then there is a serine or a threonine. So you need these 3 amino acids in a row to add on an N-glycosylation. But these sugars actually get added onto the asparagine, which is unsurprising because asparagine has nitrogens in its side chain, hence N-glycosylation. Any questions about the two types of glycosylation or anything like that? Cool. Okay. So let's now start thinking about our favorite virus, SARS-CoV-2. So there's been a lot of work on characterizing the proteins of SARS-CoV-2, especially the spike protein. And some people have done a lot of work looking at the glycosylations that happen on the spike protein. So real life example, glycosylation is real, in case you were doubting it. I would like everyone to take a few minutes and think about how might glycosylation impact the infectivity of and the immune response to a COVID-19 infection? And also go ahead and think about, would the spike protein that is produced after a vaccination be glycosylated? So take a while, maybe 10 minutes or so. Feel free to talk to a neighbor. This is a little bit outside of what we talked about in the lecture, but based on what you learned from the lecture information, you should be able to get some ideas about both of these questions. So go for it, and we'll come back together in a few minutes.
And then like you push on to your cells. So like I certain cells. Well, one is co-translation. So I think like so like it's gonna happen. Like it's, you know what I mean? Like if the protein has a structure that is gonna react with sugars, it's gonna have I think I'm I think I'm done. Yeah, I'll collect my I will say. Listen, the first week, like all of our regulars gone. They just all your regulars. All the people like Orlando was all yeah, like Cole William went down pretty significantly. A lot of dead people. Not good. And then since then, the one was people built up some type. What was that? Wow. back, bro. Not that bad, but but there's like people that are actually calling ambulance again. Yeah. Like a lot of people get COVID and they're getting so mild that they didn't have to leave their house, but now people are getting sick again. But not really like that. Um, I guess the new variants that are going around are a little bit more virulent. But it's mostly like people that are like 95 years old. With like, yeah. It's not like you know,
Y'all are being recorded, yeah.
oh boy. Yeah. There's gonna be some poor kid that's like going through this lecture. What are they talking about?
I don't think you can hear much. It mostly like is listening to me. Yeah. No, it's fine.
Yeah. There's like 170. I'm kind of young for this.
Did you have a pre-existing
Yes.
condition? I mean, everyone who's getting sick now does. It's like people for whom like vaccines don't work that well are the ones who are getting sick, which.
Um,
It is kind of sad. Yeah.
yeah, I got like CHF, COPD, diabetes, hypertension. Name something he had it. Probably extends. Well, actually,
All right, so I think it's about time to start going over this question. um, so does anybody want to start us off by saying how they think glycosylation might impact the infectivity of and or immune response to a COVID-19 infection? Go for it. Yeah, absolutely. Yeah, so it can kind of go both ways. So so like proteins, especially secreted proteins or membrane proteins like the spike protein, tend to be glycosylated. So when you are infected, you're going to see spike proteins. I guess I'm sort of maybe giving away part of the next question, but the spike proteins are glycosylated, right? So any antibody response that you make against them are going to include sugars as well as or both sugars and or proteins as part of that epitope that's recognized by a particular antibody. So if that glycosylation changes in some way, then you're absolutely right. That could potentially make it so your antibody is no longer able to recognize that epitope if there is a difference in what is there glycosylation wise. And of course, the other part of so like this is more part of like the immune response, let's say. Does anyone have more ideas of how it might affect an infection? So let's think about what the virus is doing when it's trying to infect one of your cells, right? So it's, again, you have that spike protein. It has to recognize, you wanna add on? Yeah. Right, yeah. So that would be like if the glycosylation changes or, yeah. Potentially, yeah, because you can't mount a good immune response against it. Yeah. Yeah. Right. Yeah. So that's the right idea. So like the spike protein has a part of it that's the receptor binding domain. So the spike protein has to find a particular receptor on your cells, the ACE2 receptor. The receptor binding domain has to stick to it and interact with it in the right way to actually get inside your cell. So if you have a change in a glycosylation pattern at that site on the receptor binding domain, then the virus might have trouble infecting you by getting into your cells. So does that kind of make sense? It can affect both its ability to infect you by getting inside your cells, and it can also affect your own immune system's ability to fight off an infection that's already there in slightly different ways, I guess. Yeah. Sure.
Those two have to do with the lungs, and then one would be like peripheral vasculature.
Oh, I used to know, and off the top of my head, I don't think I remember. I believe it has to do with, it's like a multi-step conversion process. So like there's like an ACE1 that makes something into something else, and ACE2 normally does the second part of that reaction, making that thing that was made by the ACE1 into the final product. And I don't want to tell you the wrong things because I no longer remember what they are. Yeah. And I think they are located in different tissue sites as well. Yeah. Yeah, exactly. It's all about location of the receptor, so it can only infect a very particular site on your body. Like it's not going to cause like a skin and soft tissue infection because no ACE2 receptor is there.
Right.
Yeah. All right, so let's now think about the second part of the question. So would the spike protein that is produced after you get vaccinated be glycosylated? So think about when glycosylation typically happens to proteins and think about what's happening when you get a vaccine. What are you doing in terms of synthesis and trafficking of protein? And would the time for glycosylation happen when you get vaccinated? It's all about time and location.
I was with the vaccine that you're with an mRNA vaccine, it's translational. So my theory was, why wouldn't it happen if you transmit the proteins in conform in such a way that if there's sugar around, it's going to react with that sugar. There wouldn't be a reason that it wouldn't be
Yeah, exactly right. Yeah, so both the mRNA vaccines and the viral vector vaccines are giving your body instructions, right? The mRNA vaccine gives you mRNA, a viral vector vaccine is DNA. Either way, it's your own cell's job to take those instructions and translate them into protein and eventually send them out of your cells to get recognized by your immune system. So that's the same exact process, literally the same exact process that we're using when we glycosylate our own endogenous proteins. It's just that the instructions came from outside of our body. But as you translate the spike protein and send it to the outside of your cell, it has to get made in the rough ER, it has to get sent through the Golgi, it has to get sent out through the plasma membrane outside of your cell. So it's going to encounter all of that same glycosylation machinery that glycosylates all your other proteins. Your body doesn't know the difference. It just got instructions to make a protein. So yes, it would be. And this is actually a good thing, right? Because when we're infected with the actual virus, it's also giving our cells instructions to translate its proteins for us and make them. And so our bodies are going through the same exact process of glycosylation and the type of glycosylation patterns that will get added, whether you're infected with the SARS-CoV-2 virus or whether you're getting vaccinated. And so this is part of what makes the vaccines pretty good, because they're pretty good at mimicking what the actual infection might look like in terms of the spike protein. Yeah. Yeah, no, it does. I think something important to consider is that when you're infected with the virus, you also have to make lots and lots of copies of the virus inside your own body to really get sick. And when you're making all those copies of the virus, you're translating all the virus's protein and you're glycosylating them. And it's the same set of instructions, the same set of RNA for the vaccine or the virus. And so your own body will make the same glycosylation patterns on both of those things. So any immune response you mount against the vaccine will be effective against those same viral proteins that you also made in your own body, right? Yeah. Yep. I mean, assuming that there isn't like a drastic difference, like in, the protein sequence that would alter like a location for a glycosylation site. Which you never know. There's a lot of variance out there, right? Yeah. So any other questions about how glycosylation might be happening when you get infected with the virus? All right. So let's look a little bit more at the glycosylation patterns in spike protein. So first we're going to look at N-glycosylation. And so this should look familiar from the lecture. This is the original 14 sugar little sugar tree that gets added when you start off N-glycosylation. And it has a lot of mannose content in it. And then as you go through the editing process, as these arrows show, you tend to have less and less mannose, and you have other types of sugars that you can tell from this little legend. And so a lot of times when people are analyzing different types of N-glycosylations, they'll often look at the mannose content, and that can tell you if it's sort of early on in the editing process, sort of more immature in a way, compared to a more like matured, more edited N-glycosylation that has much less mannose content. And so in general, people will look at sort of separate it into 3 different categories. So there's high oligomannose. And so this is more mannose, so earlier on in that editing process. There's hybrid. This is when you're trimmed down to just 3 mannoses, and then you'll add on little branches, and one of them has a fucose, and then you'll have sort of one extra arm, which isn't shown in this diagram. This diagram shows when you have 2 arms, which is a complex end glycosylation. So like here, this one has 2 arms. This one has lots of arms. These ones, here's the little fucose that gets added on. And so these are different kinds of ways we can classify the different end glycosylations. Don't stress too much about like remembering which is which. This is just so you can kind of get a sense of what people are looking for when they're looking at these. And so as people are going through, they or based on these types of classifications, people are then able to go look at the spike protein, look at all the different glycosylation sites, and classify them based on their mannose content. And so it's not clear that, or let's talk about it first. So in green, you have the highest oligomannose content. So these green ones would be like these ones, the more immature ones. And then as you go towards the hybrid and complex ones, they have less mannose. And so that's the orange and purple ones that you see on here. And so, and you can see there's a pretty big variety. There's, but yeah, but mostly I would say relatively more, or yeah, okay. I don't know why my brain just flipped them backwards. So yeah, so we see mostly purple ones, which have low oligomannose content. These means they're more highly edited, and so they're more, let's say, like mature glycosylations. And so it's sort of an interesting observation, but it doesn't necessarily have any clinical meaning. I don't think anyone that I know of is doing studies looking at like, if you change the type of end glycosylation, does that make it more or less virulent? That type of thing. But now that someone has mapped out all these glycosylation sites, that makes it easier for someone else to do that kind of study, looking at how that might have any kind of clinical effect. And then something that's also interesting to note, so we talked about before how when the virus is infecting you, the receptor binding domain has to bind to the ACE2 receptor, and how that might get affected by glycosylation. And so you can see in this picture, the ACE2 binding site is shown in blue, which is over here. And from the side view, it's up on the top. The top view is right in the middle. And you can see those sites are very no end glycosylation. They're sort of clear of glycosylation. And so that's something to keep in mind. If you did happen to glycosylate that site, that would probably have a very big effect on the virus's ability to actually get inside your cells. So interesting to note. I don't think anyone has done anything with this yet, but food for thought. So that's end glycosylation for spike protein. So now we'll take a couple minutes and talk about the O glycosylation. So for O glycosylations, there are basically 3 different types that are shown up here. They're not numbered, but so one is just made of hexosamine molecules. So those are in this blue rectangle here. So just a 6-membered sugar with an acetylated amino on carbon number 2. There are also some O glycosylations on the spike protein that have that hexosamine plus a regular hexose, so just a 6-carbon sugar with no modifications. Those can be linked together for a different type of O glycosylation. And then there's the other type, which is the first 2 plus a third, right? This one in green that's shown down here. And this is an acetyl neuraminic acid. So neuraminic acid looks a little bit like a sugar, but it has some extra substitutions on it. It's a type of sialic acid, if you've ever heard of sialic acid in other contexts, like for flu infection. Don't need to worry about its structure, but just sort of know that the spike protein O glycosylations can have any combination of these 3 components in them. So, and this is this sort of similar pattern. So like, especially when we're looking at this kind of disaccharide one at the top, this is very similar to what we were looking at for all of our other O glycosylations, because they're sort of fairly universal. So you have a hexose and a hexosamine. And then in addition, you can also be adding on this neuraminic acid. And so these people did another map, now looking at the O-glycosylation sites. And the 3 different types of O-glycosylations are shown in red, blue, and orange. And then there are also, just for information, the N-glycosylation sites, and those are shown in green. And so something that's interesting to note is that the O-glycosylations are, for the most part, right next to N-glycosylations, which is kind of interesting, right? So when you think about the amino acid that O-glycosylations are attached to, right, it can be any serine or threonine. There's kind of no rules there. But in real life, what we're seeing is that they're almost always next to an N-glycosylation. And if you remember, N-glycosylations are happening, so this is serine or threonine. N-glycosylation is happening on an asparagine, right? But it's happening on an asparagine that's 2 amino acids away from a serine or threonine. And so it's kind of cool that they were able to actually kind of map out that a lot of these serines and threonines that are right next to those N-glycosylation locations are actually where O-glycosylations are also happening. And so there's this sort of O follows N rule that a lot of proteins follow, where the O-glycosylations are right after the N-glycosylation. And that's something that we see here. Any questions about that? Yeah. Oh, so I think this is sort of showing the order. So first you have the disaccharide, and then you can have 1 through 3 of the N-acetylneurominic acids. So, yeah, I think, oh, or do you mean here? Sorry, which 1 through 3 are you asking about? This one, okay, yeah. So which one is left? Yeah, okay. It's confusing because this is also N1 through 3, but it's a different N1 through 3. This is referring to how far away it is from the N-glycosylation site, like this one, which is a little confusing. Yeah. Yeah, so just like we talked about for O-glycosylations, they start out with that disaccharide, that's a hexose plus a hexosamine, and then you have the editing process where you can add or subtract from there. Yeah. Yeah, so there's also a glycosidic bond, and that is yeah, it's also just by a glycosidic bond. So it doesn't say on here which type of glycosidic bond is. I don't know which carbon is bonding. I can look that up, but I don't know off the top of my head. All right, so if we're all tired of viruses, we can, yeah, so now we can start talking about blood types. Yay, blood types. I think they're kind of cool, actually. It's like a fun little puzzle you get to solve. So yeah, so glycosylation, we were just talking about how glycosylation is super important for immune responses and infections for a virus. Glycosylation is also really important because our blood cells have glycosylated proteins on their outsides, leading to our blood group antigens. And these can potentially cause really serious problems if you give someone a blood type they're incompatible with. So let's talk about, or first I'll let you take a few minutes and start filling out this table, and then we'll go over it together. Yeah. I actually want to just draw a line down the middle so it's more obvious that this side is for antigens and that side is for antibodies. Yeah. Yeah, I mean, and like also like this particular classification for blood and blood group antigens is like only one. There's like a number of different other sets of blood group antigens that you can use for blood. So yeah, yeah, it's not yeah. Okay, so let's get started filling out this little table. So I guess let's start with the top. So does anybody want to tell me what antigens you see in A positive and A negative blood types? Yeah, yeah, for sure, A. Yeah, so for A positive, there's the Rh factor. And then someone else has brought up a question about this, but do we see the H antigen? Yeah, so the answer is yes, but kind of. So if you recall from the lecture, the H antigen is kind of like the base carbohydrate, and then for A and B, you add on to it. So as part of the A antigen, you have the H antigen is like a piece of it. So like you probably don't have H antigen on its own, but like you do have H antigen because it's part of the A antigen, if that makes sense. And so, in fact, I'm just going to go ahead and fill out all of them all of the A's and B's kind of have the H antigen because they're all a component of H is a component of both the A and the B antigen. So yeah, so that brings us to and clearly it doesn't have the B antigen, right? So what antibodies would we see for an A positive person? Yeah. Yeah, you'd see anti-B antibodies because they don't have the B antigen. Would you see any other antibodies? No. Yeah, that's right. So change my color again. So because this person has Rh factor and has A antigens and has that component of the H antigen, you wouldn't make anti any of the other ones. So for A negative, it's like A positive, except what's the one difference? Yeah, exactly. Yeah. So it has A, but it does not have B or Rh antigen. And so then what antibodies would the A negative person be making? Yeah. B and Rh. Exactly. Okay, so let's now consider B positive and B negative. What is the main difference between A and B? Go for it. Yeah. Yeah, exactly. So B makes the B antigen, both B positive and B negative. What about oh, sorry, I put that in the wrong column. B antigen. And then which one makes the Rh factor? The positive. Yep. So then which antibodies would the B positive and B negative person be making? Yeah. Yeah, exactly. Okay, so now similar but different, let's talk about the AB people. So what antigens do AB positive and AB negative people make? Yeah. Yeah, exactly. And so then what antibodies are these people making? Right. Yeah, exactly. So if you make all the antigens, you're not making any of the antibodies. Yes. Yeah, we're getting to that. Yeah, I'll just pop it up now. So yeah, so is this person the universal donor or the universal acceptor? Yeah, so this person is the universal acceptor. All right, so then let's finish it off with the O positive and O negative. Yeah, so their primary antigen is the H antigen. Exactly, yeah, so O, people with O type blood do not make A or B antigen. They just make the plain old H antigen. So what does that mean in terms of the antibodies that they make? Yes. So O negative has antibodies against almost everything. Exactly, against A and B. So we talked about our universal acceptor, which is AB positive, because they don't make antibodies against any other type of blood. So what blood type would be our universal donor, and why? Go for it. Mm-hmm. Yeah, exactly. So they're the universal donor because they don't make any of the antigens that other people are making antibodies to. The universal acceptor is not making antibodies against any of the antigens they might encounter. Mm-hmm.
Is it kind of by, it's definitely not by design that you have antibodies against other blood types, right? So it's just kind of the antibodies that you have.
Yeah, exactly. So if you think about the way that antibodies are made, so they're made in developing B cells. As our bodies are making B cells, they are made essentially through like a random process. They're not targeting anything specific. You just make a whole vast variety of them. And then as they go through the process of maturing and developing, your body basically tests them out against your body, and it deletes any B cells that react against yourself. And if it doesn't see them in yourself, it holds on to them because it's something foreign or something that's not yourself that could potentially be bad for you. So in this case, it's kind of an accident and not really ideal, but it tracks with how your immune system works because it's not yourself. Therefore, it's bad. Yeah. Yeah, so where's that slide? Just gonna go back. Sorry, don't look at the screen flashing. Where's that slide? There it is. I was thinking of, what was I thinking of? Yeah, so the H antigen, I thought there was a better picture on here. Maybe I'm losing my mind. Yeah, so the H antigen is kind of the core part of this sugar structure that's put on the protein. And then A and B have just one sugar added on to that core H structure. And so they each have the same, I should put this in a different color, but A and B contain that same core structure that's part of the H. Yeah. 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 Right. That's really interesting. So how can you make A or B antigen if you can't make H antigen? So you don't, oh, okay, you just don't make any. Okay. Cool. Okay. I've never heard of that before. Yeah. Yeah, so that person would not be able to receive anyone else's blood. Yeah. That's wild. That's really interesting. Cool. Thank you. Yeah. 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 I mean, I wouldn't ask it in a way where, right, if you had to say, yeah, I guess I'm trying to say, like, I wouldn't ask you a solid yes or no. I would give you a way to say, like, it's there, but it's part of the other antigen. Yeah. Yeah. Yeah, and I think the chart is kind of designed this way to make you think about where the antigens actually arise from and the fact that they all arise from that original H antigen. Where were we? Okay. Any other questions about blood type? All right. So in that case, These next couple of slides are not part of your exam. They weren't really part of the lecture material, but they might be interesting. They're sort of a real-life example of glycosylated proteins, or a process that's used to study glycosylated proteins. So the Nobel Prize last year in chemistry was awarded to these people who worked on click chemistry. And click chemistry is a technique that's used for studying glycosylated proteins in the lab a lot. So there's, I think, 3 or 4 slides in here that if you are interested, you can go through them. I won't read them out to you. If you're interested, please look at them. You can come chat with me about it if it's something interesting to you. If it's not interesting to you, don't look at it. So that's these. And that's all I have on carbs. So it's a little bit early. I have your exams, exam 1, if you want to take a look at your exam 1. And other than that, just remember your quiz 6 is due tonight. Quiz 7, which is on this chapter, chapter 8, is going to be due next Thursday. It should be opening up in a few minutes. And and next week we'll do chapters 9 and 10, and you'll be ready for your next exam. So have a great weekend.